So, what I thought we will do today is to look at two examples, okay, and two examples from biology, of course, but I chose one specific type of system, which are the cytoskeleton and motors, since we have talked a lot about DNA and bacteria and so on. Maybe I have not talked, we should learn some other biology as well. So, the plan is to sort of introduce what is this cytoskeleton and what are motors, and then just look at two very simple models for these using this language. Cytoskeleton and motors, right. So, this is what we will talk about today. Okay. So, here is an image of a cell, okay, with certain things stained. So, what is stained are three things. One is DNA, the other is a type of filament called microtubules, a type of filament called microtubules, and another is another type of filament called actin. So, this is microtubules are in green and this actin is in red and DNA is in blue. Okay. So, as you can see, so these are many cells, of course. So, this is for example one cell over here. What you can see is that the DNA is of course inside the nucleus. So, the blue part is over here. You have this sort of filamentous network essentially of these green type of filaments, these microtubule filaments. So, essentially if you have a cell, you have your nucleus over here which has all your DNA, but outside you have this dense network of filaments, okay, which are these microtubule filaments, which is what is shown over here in red. Okay. And then at the, of course, there is also in the bulk, but mostly in the edge of this cell are these other sorts of filaments, which are my actin filaments. So, mostly, so this actin filaments will mostly mark the edge or the boundary of each cell and close to the boundary. So, things network. So, this is two types of networks. One is a network of polymers called microtubules. The other is a network of polymers called actin. These are proteins. So, these are macro molecular assembly. So, these are made up of many proteins repeating to form this sort of form these polymers. But like we had said, the inside of the cell is very dense, and of course, it is much more dense than this. This is only two things labeled, but you can see that these microtubules and actins form this very dense network inside the cell. They have two different roles. So, okay, first let me show you about microtubules and then I will show about actins. So, here is what a microtubule looks like. A microtubule looks like a hollow cylinder. It looks like a hollow cylinder. Okay, that is the cross sectional view. And this is the length of the cylinder. It is made up of a protein which is a dimer, which is called an alpha tubulin. It is made up of a protein dimer called alpha tubulin and beta tubulin. So, this together makes up my protein dimer. So, that is why when you see this tube, you will see there is a green color and a blue color. Then what happens is that you stack these dimers on top of one another. So, alpha beta, another alpha beta, like that, to make a single filament. This is called as a protofilament. Okay. Then you take multiple such filaments, and these multiple such filaments wrap together to form a cylinder. So, this is one protofilament. This is one protofilament. This is the second protofilament. So, each of this has this alpha beta structure. Okay. Is this clear? So, it is a hollow cylinder. The hollow cylinder is made up of I don't know, straws or pipes, if you will. And each of these straws or these protofilaments, as they are called, is made up of this repeating dimers, this alpha beta tubulin heterodimers. Now, these microtubules are fairly stiff. So, what that means is that the persistence length of these microtubules, remember when we are talking about polymers, we talked about persistence length where the tangent vector decorrelates and which measures the stiffness of this polymers. And these persistence lengths can be of the order of hundreds of nanometers or microns. So, which is why I've drawn these as sort of straightish rods, which does not mean they cannot bend. Other proteins can come, they can change the rigidity of these polymers, but in general these polymers are fairly stiff. So, they are essentially like rods, and what this does, one of the major roles of this microtubules is to sort of form this network essentially, and this network serves as a railroad or a transportation network inside cells, in that, as I will show, these serve as highways along which different proteins can move from one point of the cell to another. Okay. Instead of doing a random diffusion, so if you had a protein which was produced here, let us say inside the nucleus, and then that protein is needed, let us say, on the membrane, right? It has to get from here to there. Now, it could do diffusion, of course, that is always a possibility, but like we realize, these insides of the cells are fairly crowded environments. So, if you have to go from here to there through diffusion, that would take fairly long amounts of time, even for normal cell sizes, since especially given the level of crowding. So, generally what it does is that it hops onto one of these tracks, and then these tracks are connected to one another. So, it is like a very dense metro system, and then this cargo goes on these tracks to wherever it needs to go. Okay. So, the track suites, the microtubules, these microtubules over here. So, that is one of the main functions. The microtubules also provide structural rigidity, which is why it is called cytoskeleton. But one of the main functional roles is to sort of act as a transportation network through which molecular cargo can be transported inside cells. Okay, fine. All right. Now, you see this microtubule. So, this whole thing is my microtubule often. Okay. And you see this is a polar structure. This is a polar structure because of this polarity, not in the sense of an electric polarity, but in the sense of a chemical polarity, which means that if I were to look at this end of the microtubule, so I look at this cross section, what I will see is a ring of alphas, right, alpha, 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 alpha. If I were to look at this end, what I would see is a ring of betas, beta, beta, beta. So, therefore, the two ends of the microtubules have different properties from one another, because you have alpha exposed in one end and you have beta exposed in the other end. Okay. So, these polymers are chemically polar, they are chemically polar. So, they have a chemical polarity. After this beta, there will be of course another alpha, but what I am saying is that you have a pipe, right. So, one end of the pipe you will see alphas. If you cut it, of course, so I do not know, maybe you are saying if I cut it like this, if you break it like this, yes, then of course you will see alphas, but because these always come in heterodimers, these bonds anyway are more difficult to break. So, even if you were to snap a microtubule, when something cleaves a microtubule, you would generally get these. So, this dimer comes off as a whole essentially. So, one end is always going to be generally betas, the other end is always generally going to be alphas. So, it has a chemical polarity, and this is one end is then called as a plus end and the other end is called the minus end. I just say I emphasize it's chemical because sometimes it gets confusing. There is nothing electrical polarity about this. One of the key features of these microtubules is that these are dynamic objects, in that the length of this cylinder is not fixed. Okay. So, I have this microtubule, the length of this microtubule is a stochastic quantity, it fluctuates, and what that means is that this microtubule in certain states can polymerize, at some times it will polymerize, which means that this length will grow. Okay. So, it gets in a growing stage. At other times it can depolymerize, which means that the length will start to shrink. So, these transitions are called catastrophes and rescues. So, if you go from a polymerizing state to a depolymerizing state, that is called as a catastrophe. If you recover back from a depolymerizing state to a polymerizing state, that is known as a rescue. So, this just shows this sort of thing. This is the growth phase when it is growing, this is the shrinking phase when it is shrinking. And then if you look at lengths of microtubules. So, for example, this is an experimental plot of microtubule lengths as a function of time. You will see that you have growth. So, this is the growth phase when the microtubule is polymerizing, the microtubule grows. At some time it shifts to this, it undergoes a catastrophe. So, it shifts to this depolymerizing or shrinking phase, and then suddenly it depolymerizes. So, a couple of things. Of course, one is that so this is the stochastic process. So, one of the things to note is that the growth velocity is not the same as the shrinking velocity, right? Growth happens over time scales of many seconds, hundreds of seconds even. Shrinking typically happens over very few seconds. So, the shrinking events are very, very sharp. Okay. So, once it transitions to this depolymerizing state, these subunits just fall off very quickly. So, the length of the microtubule shrinks very fast. This behavior is called the dynamic instability of microtubules, where microtubules grow and shrink. No, it depolymerizes. So, it does. So, breakages can happen, but this process is not a breakage. This process is that I have these protofilaments, and with these alpha beta tubulin dimers, these just start to come off with some rate. Okay. So, these start to fall off as this. So, that is what is shown over here. These subunits start to fall off. As it starts to fall off, the length goes. Yes, exactly. So, sequentially. So, it's not that again it's not that you have this protofilament. It's not that generally it's not that dimer from the middle starts to unbind. It unbinds from the ends itself. So, it is, but the process is very, very fast. So, those off rates in the shrinking state are very high, which is why you just see this very sharp decrease. So, this is what it looks like. So, here is an experimental image of microtubules. So, these long things are the microtubules, and hopefully you will see that they are growing and they are shrinking, they are growing and they are shrinking. Okay. So, this is a feature. It's not always in such highly dynamic phases. Depending on the cell cycle, it can also the cell adjust the dynamicity, but in certain phases these microtubules can be very dynamic. They will grow and shrink and grow and shrink. So, I don't think I said. So, I said these are made up of many protofilaments. Typically, a microtubule is made up of 13 protofilaments. There is of course a distribution. Some might be 12, some might be 14, and so on. But generally it's very sharply peaked around 13 protofilaments. So, 13 of these filaments form a cylinder that gives you one microtubule. And like you see, the length can be of the order of microns, which is essentially of the order of cell size, right. So, looked at this, look at these microtubule lengths. These are of the order, the y-axis is of the order of microns, which means that these microtubule networks can span essentially whole cells. Clear? So, this is a brief intro to microtubules. Next, I will talk about this other filament, which is actin filaments. So, these are actin filaments. Just like this is made out of this protein, tubulin protein, actin is made out of actin protein. So, this is actin, and in particular when it is in solution, it is called G-actin or globular actin, to sort of distinguish it from this filamentous structure, which is filamentous actin. Okay. Again, when actin polymerizes, you get many of these G-actins coming to form one filament. Okay. Two of these filaments wrap around each other to form this actin. Okay. So, this actin is like two filaments wrapped around each other, in two helical filaments wrapped around each other. Each filament contains this G-actin, G-actin, G-actin. And again, this also has a polarity. So, microtubules are 13 protofilaments, actins are two filaments. This also has polarity, and here the polarity is like a structural polarity, in that the proteins have a structural asymmetry. So, when you have this globular protein, it is not really a sphere. The canonical sort of cartoon is to draw something like this, and then the next one is something like this, right. and then you have one end which is called as the barbed end and the other end which is called as the pointed end, and then these again correspond to a plus and a minus. So, when you have this filament something I am drawing very badly, but you get the idea from this cartoon. So, there is a structural asymmetry of this G actin, which means that when you assemble into filaments, again if you look from one side you will see some structure like this, if you look at the other side you will see a pointed structure like that, and that gives this actin filaments some polarity as well. I forgot to say for microtubules, but these polymerization processes are essentially require consumption of energy, and the way this happens is that so when you have this globular actin in solution, it comes bound to an ATP molecule, and ATP molecules are the energy currency of the cell, right. When these bind to a filament, when this globular actin comes and binds to a filament, this ATP gets hydrolyzed to ADP. So, ADP plus phosphate, and it releases energy, and that energy sort of goes into stabilizing this filamentous structure. Similarly, for the case of microtubules, when you have this tubulin dimers in solution, when you have this alpha beta tubulin dimers in solution, they come bound to a molecule which is like ATP, but instead of ATP it's GTP, guanosine triphosphate. And again, once it's bound, it hydrolyzes, it produces GDP, and that energy goes into stabilizing the microtubule structure. So, these are this polymerization processes are in that sense non-equilibrium, in that they are maintained by a constant influx of energy through this ATP to ADP or this GTP to GDP reactions. So, like microtubules show a dynamic instability, actins are also dynamic, but they have a slightly different form of dynamicity which is called as treadmilling, which is called as actin treadmilling. Okay. And what happens is that let me draw an actin. Okay. So, this is my plus end, this is my minus end. Because you have this structural asymmetry, you have plus ends and minus ends, and of course, you have these things floating around in solution, right. So, these are your G actin floating around in solution. These can bind, and whatever is bound can unbind, of course. So, you have an on rate and you have an off rate, right. But because there is a structural asymmetry, there is a polarity, these on and off rates need not be symmetric in the two ends. So, for example, I could write that this is K on plus, this is K off plus, whereas here the on rate is K on minus and the off rate is K off minus. And these two need not be equal. In fact, they are not equal as a consequence of this polarity. Now, depending on how these rates, which one is greater, which one is smaller, and so on and so forth, you can get different regimes. But generally, actins will often exist in a regime where essentially you are going to add monomers to the plus end and take away monomers on an average. Of course, these are stochastic processes. So, everywhere it will add and fall off, but on the average you will tend to add monomers to the plus end and tend to detach monomers from the minus end. Okay. So, this is called as treadmilling because it's being added here and it's falling off over there. So, this is called as actin treadmilling. It's a different sort of dynamicity as compared to microtubules. Actins have again many functions. Two of the major functions is that it generates contractions, and I'll explain maybe how. And it also so it generates contractions, and it also is responsible for cellular locomotion. So, this is a video that shows locomotion. So, here is a cell with actins labeled, and what you will see is that this cell sort of extrudes protrusions which are called lamellopodia, and these lamellopodia are sort of So, let me first draw and then you can see. So, the cell will extend protrusions called lamellipodia, and in these lamellipodia you will have very high concentrations of actin, and these actins through this treadmilling process generates forces which elongates this lamellipodia. So, the cell can send out multiple lamellipodia in different directions to sort of sense the environment. Then once it chooses a direction, there are other proteins that sort of attach this end to a substrate, and then the cell can. So, it's essentially I extend a lamellipodia. If I know that this is the direction I want to move in, I sort of grab onto the substrate and pull myself in. So, it's in a very simple. So, for example, here is one very long lamellipodia. Here is another, and you'll see that at this leading edge there are high concentrations of actin which sort of drive the protrusion of this lamellipodia through this sort of a dynamic process where one end is growing and the other end is sort of shrinking. So, having talked about cytoskeletons, now I said that for example, things go on these tracks, for example, microtubules, also actin, there are things move on actin as well. But how does something move? Something moves on this track essentially when it is carried by a different class of proteins, and those proteins which help to move objects or what are called as cargo from one end of the cell to another, those proteins are called called motor proteins. Okay. So, I will just give again a brief introduction to molecular motors. The simplest thing, essentially any protein is a molecular motor which takes in chemical energy and uses that energy to do mechanical work. Okay. That is like my basic physics definition of a motor. There are various classes of motor proteins. Here I will just talk about essentially motors that move on these actins and microtubules, but those are not necessarily the only sort of motors. So, for example, even the RNA polymerase which moves on DNA, that is also a motor in that is also active. It takes in energy and it produces this RNA strands and so on. But for today, I will just talk about these class of motors which walk on these microtubules and actins. Okay. So, here's many of you may have seen this. This is a very famous cartoon in that here it's this track over here is your microtubule. So, you will see this sort of a dimer structure, this repeating dimer structure. Over here is a big vesicle. So, it is a lipid vesicle which has various proteins and stuff inside it, and what the protein that attaches this vesicle to this track, this microtubule track, is a motor protein. In particular, what is represented here is one particular type of motor protein called kinesin, and what it does is that it has a head domain and a tail domain, and these two head sort of subunits, it walks on this cargo. So, it walks on this microtubule. So, you have this alpha beta structure, here is your motor protein. It has two heads like so, and then when one head attaches, the other one can detach and make a step to the other side, and then this one detaches and makes a step. So, it is a repeated chemical binding and unbinding events, but essentially what it translates to is motion of these motor proteins on the underlying filament. All right, and there are various types of motor proteins depending on various things. So, for example, myosin is a motor protein that walks on actin, and again this is a broad family. There are different subclasses of myosin, but broadly myosins are motor proteins that walk on actin. Kinesins and dyneins are motor proteins that walk on microtubules. Okay. And the difference between kinesin and dynein is that kinesins walk towards the plus end. So, again because these filaments are polar, the motors can also recognize polarity. So, if you have a plus end and a minus end, if you have a kinesin, then that preferentially moves towards the plus end. If on the other hand you have a dynein, that preferentially moves towards the minus end. And there are various ways to measure step sizes, speeds, forces that you exert, and so on and so forth. So, the different sort of physical properties of this motor proteins can be well characterized through experiments as well. Alright, how do you generate forces? So, I said that you can use these motor proteins to generate forces, and the way you generate forces is that motors not only carry cargo, but they can complex and they can bind two microtubules. So, let us say you have a microtubule, what orientation have I drawn over here? Plus plus or something like this. Okay, so you have one microtubule where the plus end is over here, the other so antiparallel microtubules essentially like this, and then let us say you have very bad drawing, but you have this dyneins, maybe a dynein complex which binds these two microtubules. Okay. Now, dyneins will want to walk towards the minus end like this, right. Maybe I should try to draw one configuration like this. So, this dynein will want to walk towards this end, this dynein will want to walk towards this end, and therefore, they can generate either positive or negative forces for overlaps, okay, depending on what orientation I have drawn my microtubules in, and whether it is a dynein attached or a kinesin attached. Okay. So, they can cause sliding of microtubules, or if you do not allow sliding, in that if you have some other proteins which do not allow sliding, which are these linking proteins, then they can bend these microtubules, because they still want to walk towards the minus end. So, they can bend these microtubules, and in that way again, so they can cause so this motion of these motor proteins can cause sliding, or they can cause bending as well, and in this way they can generate forces. Okay. I just wanted to, maybe I will not show this, but just to say you can look at this up on YouTube. So, when you contract and expand your muscles, essentially this is what is happening. Motor proteins are walking on filaments. So, in particular for muscles, it is myosin which is walking on actin filaments, and these force generation, this contraction and expansion, or the sliding of these filaments, is what generates, is what causes these muscles to flex and unflex. So, here is another particular example. So, this is during cell division. These green things are microtubules, and during cell division, of course, you know that you have your DNA, and your DNA. So, each chromosome has 2 copies, and when you are having cell division, one copy needs to go to one daughter cell, another copy needs to go to another daughter cell. And the way the cells separate out these chromosomes is that these microtubules sort of polymerize and depolymerize until it latches on to one of these chromosomes, and then it depolymerizes and it pulls the chromosome back. Similarly, so there are 2 spindle poles generally. So, these are called spindle poles, and then these microtubules can sort of through this dynamic instability can separate out the chromosomes. Further, after the chromosomes have been, so here is a cartoon of that. So, these are the 2 spindle poles. The microtubules polymerize from the spindle poles until they find your chromosome, and then they pull them apart. That is one phase. And once they are pulled apart, the cell needs to constrict so that the cell can divide, and that again happens through contractile activity of the actin and myosin filaments. Okay. So, these interactions of motor proteins with microtubules is responsible for a lot of underlying cellular processes, transport, force generation, locomotion, cell division. So, these are sort of ubiquitous. So, it is good to know a little bit about these systems as well. All right. So, that's the sort of biology. I don't think I have. Okay. Let me show 2 more things before I do. So, how do you visualize them in outside of cells? So, there are 2 sorts of assays that are possible. One is called as a gliding assay, and the other is called as a motility assay. So, a gliding assay is something when you have, let us say, a cover slip, and you place many motor proteins on them. Okay. And you fix these motor proteins. Okay. So, you tether them to the surface, whatever is your cover slip, and then you place these filaments, let us say microtubules. As the motor proteins try to walk on these filaments, the motor proteins themselves are bound, so they cannot move. So, what they cause is that these filaments start to move. Okay. All right. So, if you look at these sort of gliding assay experiments, you will see that these filaments are moving, but these filaments are not self-propelled in some sense. These filaments are moving because there are motors on this surface which you cannot see, but these motors are pushing these sort of filaments, causing these filaments to flow. When you put, so this is of course a very dilute dilute limit of these filaments. If you put a very highly dense system of filaments, you can see various interesting things. So these are called motor driven active flows. So here is something where you have again microtubules and kinesin proteins. The microtubules can move, the kinesin proteins are trapped depending on how much ATP you put. So which means how fast or how much your motors can move. You get these very nice different flow patterns. So these are various vortex patterns that emerge in these motor microtubule sort of mixtures. Finally, the other sort of experiment that you can do. So here you immobilize motors and you let microtubules move. On the other hand, you can have microtubules fixed and have cargo which can be either some biological cargo or a glass bead or something, and then you attach molecular motors to this cargo which can then walk on the microtubules. As they walk on the microtubules, they pull the cargo, but because you are in an optical trap, the optical trap exerts a restoring force, which means the further you walk, the more forces you feel, and ultimately what you will get are traces of this position of the cargo as a function of time. So this is what that would look like. So this is what that would look like. So here it's actually a cargo like this, a bead like this, which is moving on a microtubule, and you can't really see the microtubule, but there is a very faint, if you look, maybe not here, but if you look very closely, there is a faint line over here, which is a microtubule, and the cargo is moving on this microtubule. Might be a little too much biology for you, but think anyway. So we'll use these terms again and again, and at some point, hopefully, you'll get used to these terms. But having said that, let us now come back to some simple physical problems that we can solve using these systems. So I'll discuss two papers, one PRL and one PNS. So I talked about dynamic instability of microtubules, right? So let me talk about a model for dynamic instability. And this is, I think, 1990 something, PRL 93. This is one of the classic papers of microtubule polymerization and introduces a theoretical model for this dynamic instability. Remember, what is dynamic instability? When you have microtubules which are growing and then shrinking and then again, that curve is a fat curve. It's growing and then it's shrinking and then again growing and then again shrinking and so on. Okay. So this is dynamic instability. And basically what this tried to ask within what this paper tried to ask, and again, there are much more complex models out there. But what this very first and very simple model tried to ask is that if I have these microtubules, which can be in a growing phase, which can be in a growing or a polymerizing phase, or in a shrinking phase, or a depolymerizing phase. So this is polymerization, and this is depolymerization. Under what conditions will I see some sort of, so you could imagine two sorts of scenarios, right? So if I looked at length of microtubules as a function of time, right? One could imagine two sorts of scenarios where it grows and then it shrinks and then again grows and then again shrinks and again grows and again shrinks, so on, right? Something like this. Or in principle, you could think of a scenario where you shrink, but then you again grow and so on. In that, if I were to look at the average length of these microtubules, maybe in one case I would see something like in spite of the dynamic instability, some sort of a mean constant average length, and in the other case I could see maybe a growing sort of a microtubule. Okay. So this is called a bounded growth versus unbounded growth. And what this model tried to understand is when would I get these sort of bounded growths versus unbounded growths using a very simple physical picture. Okay. All right, is the question? So, bounded means when this average length is sort of finite, and unbounded when this length continues to grow with time. All right. Now, this system is clear. Okay. So, from now on physics, okay, but anyway, guided by the biology. So, the microtubule exists in a growing stage, or it exists in a shrinking stage, right, in a shrinking phase, right. So, I could say that I could write two types of probabilities. One is a probability for a microtubule to have n subunits, so n tubulin dimers, and exist in a growing phase, or a microtubule to have n subunits and exist in a shrinking phase, right. And I could write down the master equation for these probabilities. Right. I could write down in principle the master equation for these probabilities. To write down the master equation, I need to think about what sort of processes are possible and what are the rates of these processes. So, let me just write that here. So, again I have a growing phase and a shrinking phase. So, this is my plus state, this is my minus state. Okay. When I am in the growing phase, I can do two things. I can add a subunit, right, and therefore increase the length, and let me say that that rate at which I add subunits is V plus. Okay. Or if I could undergo a catastrophe and switch from the growing to the shrinking phases with some rate. Let me call that F plus minus, which means I was at plus, I go to minus. When I am in the shrinking phase, I can lose subunits with some rate, which is V minus, let us say, and or I could transition to this growing phase, which is F minus plus. So, this is my simple, or this is by two people called Dr. Ohm and Leibler. So, so this is their simple model for dynamic instability of microtubules. You have two phases, and here are your appropriate rates. All right. So, I want to write down the master equation, the forward master equations, which means I want to write down the gain terms and I want to write down the loss terms. Okay. So, who is going to tell me? Siddhant, what are the gain terms? Tell me one gain term. So, how do you get to a state where you are in the growing phase and you have n subunits? From what state can you get here? Okay. So, I would write P of n minus 1 in which state? Plus state. And what would be the associated rate? F minus to plus. What is this F minus to plus? It is the rate at which you convert from shrinking to growing, right? But that is not what is happening here. You were in the plus state, you continue to be in the plus state. So, what is the correct rate to write here? V plus. All right. What is another? Are there any other gain terms? What is it? F minus plus P, P n minus. Very good. Okay. And you will, so these are your two gain terms, and then you will have the loss term. The loss term is that you were here P n plus, and you have grown. So, you have come out of this state, and similarly you were in this growing state, but you now undergo a catastrophe to switch to the shrinking phase. So, you have that. All right. And similarly, the gain term over here is a V minus P of n plus 1 because it's the shrinking phase. So, P of n plus 1 comma mine in the minus plus that term F plus minus P n plus minus V minus P n minus plus F minus 2 plus, no, sorry, this is the lost term I am writing. So, minus F minus 2 plus P n minus. Is this clear? Of course, you will notice that in some sense it is very similar to the run and tumble master equations that we wrote down, right. You had two states, run and tumble. In each state you had some rates of adding or subtracting, you had rates of switching, but these are very different physical processes, of course, but the mechanism and the machinery is very similar. Okay. All right. So, here is my master equation. I can do some manipulations on it, which is that I could expand these n minus 1s in some in a Taylor series. Okay. So, then I would get, so P n minus 1 would be P of n comma plus minus V plus del P del n, right. So, I had P of n minus 1 plus this I am writing as P of n plus minus 1 into del P del n of P n plus. So, I just do a Taylor series expansion. Once I do a Taylor series expansion, this term cancels out. Similarly, here if I do the n plus 1, if I do a Taylor series expansion, I would get a V of P n comma minus plus V minus del P del n of n minus, and again, these are discrete updates, of course. So, we are taking a discrete. So, this is an approximation in that this is the polymerization model, right. So, when you are adding things, you are adding subunits, which is an alpha beta subunit. So, you cannot add continuously, and this subunit is roughly 8 nanometers. So, this 1 essentially means 8 nanometers, but on the other hand, remember that my microtubule lengths are of the orders of microns, a few microns. So, in that sense, this delta x is much smaller compared to the L of the microtubule. So, I can do a continuum approximation. But yes, underlying it's a discrete process. All right. So, therefore, I have, once I have expanded this, so let me just write this little bit of a condensed notation. So, I have del T of P plus is minus V plus del n of P plus F minus plus P minus. This is I am dropping the n subscript. So, this is P plus F minus plus P minus F plus minus P plus, that is one equation, and then del T P minus is equal to plus plus V minus del n P minus F minus plus P minus plus F plus minus P plus. So, I get this equation for the probabilities of the microtubule to be in the plus state and the minus state with n subunits. Okay. So, up till here is in some sense stochastic process. After this, it is up to you how you solve it. So, let me say that we will solve it, or rather what these people did is that they solved it in the steady state. And again, in the steady state, what it means is that, because remember what I want to know is that whether I have some average length which is constant, or whether I have some growing average length. So, I look at the long time limit. So, if I look at the long time limit, this time derivatives I set to 0. Okay. Which means that essentially, if I am solving for these probabilities, what I am solving is this equation del n p plus del n p plus is minus and then minus. So, f plus minus by v plus, sorry, this I have brought to that side, that is a minus. So, there is a minus p plus and then a minus f minus plus by v plus p minus, and then I have del n p minus. So, del n p minus, I have a minus v minus over here, which is f plus minus by v minus p plus, and plus f minus plus by v minus p minus. So, in the steady state, if I want to solve, which one is plus? Sorry, first equation, second term. Okay, f minus plus, yes sir, plus. Thank you. Which means that if I am solving for these probabilities for the microtubule to be in the growing phase or the shrinking phase in the steady state, I need to solve these equations, right. And again, how you solve these equations is up to you. Easy way to solve is to just realize, just write this in matrix form, right. So, this is some del n, some p matrix is some A matrix times the P matrix, where A is this 2 cross 2 matrix minus F plus minus. And then once you have solved it, this is like an exponential solution. So, you get this matrix A, which is whatever minus F plus minus by V plus, F minus plus by V plus, F plus minus by V minus, and then F minus plus by V minus. Get the eigenvalues of this, get the eigenvectors of this, and then combine them to solve for P plus and P minus. So, again, the differential equations you should be able to solve. So, this I will not do. Let me just write down the solution. I have the eigenvalues somewhere. This even the paper will not do. So, you will have to actually do and solve and check. So, if I do this, I get the eigenvalues of this matrix as one eigenvalue is 0 and the other eigenvalue is F minus plus V plus minus F plus minus V minus by V plus V minus. Okay. And then you find the eigenvectors corresponding to that, and you can show that that then P plus of n is A F minus plus by F plus minus e to the power of lambda 1, which is 0. So, e to the power of 0 is 1 plus B V minus by V plus e to the power of lambda 2n, where lambda 2 is this, and similarly V minus of n is A into e to the power of 0 plus B into e to the power of lambda 2. This one I thought is a minus, no? So, maybe I have made a mistake in the signs, you can check. I may have a somewhere, sign mistake somewhere. So, that you can check and do. Okay. The essential thing is the same, but this eigenvalues I am fairly this is the correct eigenvalues. And once you have bound, so again my question was, when will you have bounded growth versus unbounded growth, bounded versus unbounded. You can see that you will have bounded growth when this lambda 2 is negative, right. So, and you can work that out as well. So, for example, you can calculate what is the average length of your microtubule, right. What is the average length of your microtubule? It is sum over n times p plus of n plus p minus of n, right, in principle. So, you could calculate what is the average length of your microtubule, but in general, so you can do this and show, but here from here also from this set you can see that for bounded growth what you need to have is lambda 2 has to be less than 0, which enforces some constraint on these very biological parameters, that minus plus v plus has to be less than f plus minus v minus. And these are, so what it tells you is that if you have this, if you have to have this catastrophe and shrinkage events such that you have in the long time limit some sort of a steady length, then you must have some sort of this sort of a relation between your switching frequencies f minus plus, f plus minus, and your rates of growth and shrinkage. Okay. But these are things you can measure experimentally. For example, if you have a plot like this, you can measure what is the rate of growth, which is this slope. You can measure what is the rate of shrinkage, which is that slope. You can measure how many times it switches from this state to that state on an average. That will give you f minus plus and f plus minus, and you can see that essentially whether your biological microtubules lie in this phase or that phase, and it mostly turns out that it lie in this bounded growth phase where you will have some sort of steady state length distribution. So, even though you have this dynamic processes of growth and shrinkage, at the end of the day in the steady state, these microtubules operate in a regime. The rates are such that you operate in a regime where you have some sort of a steady length scale. Essentially, so one way is this, of course, you work out this average n, and then you say that this average n has to be finite, okay, and then see what you will get this result. The other way is that you can always just see that. So, you need to find what are these constants a and b, right, which means that I need to impose some boundary conditions. So, if I want bounded growth, what I need is then that the probability, whether plus or minus does not matter, as n tends to infinity must go to 0, okay. And of course, I must have normalization, which means that sum over p plus plus p minus must be equal to 1. With these two a's you can, so this essentially says that these a's must be 0, okay. So, these a terms drop out. What you have are just this, okay, and you can find out what is b in terms of using this condition. But then again you have to have n going to 0, which means that this lambda 2, the sign of this term has to be negative, right, because otherwise as n goes to infinity you would not have that going to 0, which means that this lambda 2 term must be negative, which means this condition on the rates. So, you could do that as well. Anyone else? I thought I will do 2 problems, one for these filaments and one for the motors, but I can't do the motors. So, yeah, but this thing you can ask me if there are any doubts. Over here, yeah, we are approximating linearly. Well, that is true, of course. These are generally fairly good approximations to work under, as long as you are sort of looking at length scales where you know, length scales and time scales, where this delta x, which is 8 nanometers, is essentially smaller than L, or whether your delta t's, which is your rates at time scales at which you add or remove a monomer, that is smaller than the times at which you are looking at these processes. So, again, so for example, if I were to think of the Fokker-Planck equation, you have a drift term, you have a diffusion term, but you have also these all higher order terms. But for most processes, these terms, the higher order terms, are much negligible. All right. Okay. So I'll stop here.